Hello toppers, how are you? I think you are feeling well. Today I am going to discuss about the cell cycle, the important one chapter of third unit of 11th class. Here, cell cycle phenomena was previously described by Rudolf Virchow. He gave a sentence that is termed as omnis cellula e cellula. The year was 1855. So Rudolf Virchow gave a sentence omnis cellula e cellula means the formation of the new cell is due to the division in the pre-existing cell. So the formation of the new cell is due to the division in the pre-existing cell. This was given by Rudolf Virchow a long time back. Now the cell, what is cell cycle? Cell means structural and functional unit of life. Then cycle means changes that taken place from the starting and completes from the that end starting point, and that is the end point also. So this is termed as cycle, and there will be some changes or series of changes. But here, in case of cell cycle, the series of changes will be in a genetically controlled and coordinated manner. Coordinated manner means one by one. So, the thing will happen like this. Next, the scientist was who gave this phenomena in a description way was Howard and Pals. Howard and Pals in 1953 he gave a definition of the cell cycle suggests that cell cycle is a genetically controlled series of changes that taken place in a newly formed cells by which it duplicates its genome, synthesizes its constituents, grows in size, and divides to form the two sacs. So this definition came in origin. The scientist was Howard and Pals. These genetically series of changes will be in the coordinated manner, and this is genetically controlled series of changes. So now the definition will be well defined. So now we got it, the genetically controlled series of changes, those are in a coordinated manner, came in origin and formation of the cycle in a cell that comes to originate of the second cell. So division from the pre-existing cell will form the next cell. So go on, the progeny will be formed like this in an embryo cell, in a meristematic cell, in a germinating cell. So this type of cell formation will take place due to this cell cycle. So the chapter is cell cycle and cell division. First, we will discuss about the cell cycle overview and these changes, the changes those are will have those happen in the cell, and after that we will discuss about the division, that is mitosis division and meiosis division in a description way. So Now definition is clear. Okay, so these changes, genetically controlled series of changes, when taken place in a cell, the formation of the new cell will be there. The what time it will take? The thing is, how much time will it take to form a new cell? That time, the formation of the new cell from the existing cell, the time interval between this is termed as generation time. That is termed as generation time. Generation time. And this varies from cell to cell and the environmental condition also. How? The environmental condition will be favorable. The time or generation time will be in a favorable condition, it will be good, optimum range. When unfavorable, the time taken in a formation of the new cell, it will be very long. So the generation time or time interval to form the new cell will be different in case of cell to cell and in the environmental condition also. So the time for or generation time for human cell, in culture it is taken for human cell, it is 24 hours. In case of yeast, it is 90 minutes. And in case of bacterial cell, it is 20 minutes. So you got it, my points? The thing is that generation time vary, but in culture we have taken the human cell 24 hours, yeast 90 minutes, and bacterial cell 20 minutes. So time varies from cell to cell and the environmental conditions also. Next, this cell cycle has many series of changes. Those are classified into two stages. Like first one is interphase, second one is M phase. This interphase, long phase, it is time taking phase, and it is the preparation, it is the actual preparation. Here the preparation will take in place, and here the formation of the new cell will take place. So what is that? It is the actual divisional phase. Actual divisional phase. And first one is, what is that? It is the actual preparatory phase. Now this is the time taking, I have told you. So what time it will take? See, see, interphase, it is also termed as intermitosis. Why intermitosis? First, see, intermitosis. It is termed as intermitosis because the series of changes that taken place in a newly formed cell before it is the capable of the new divisional phase, means that when it is capable of new changes, cell will rest or preparation will take place, it is termed as interphase. That is why it is intermitosis. Between two divisional phases, that is come in origin, termed as intermitosis. Between two M phases, interphase will come in origin. That's why it is termed as intermitosis. It is energy phase, time taking phase, because energy, so much energy is required in phase. It is a preparatory phase, time taking, because 75 to 95% time consumes of the generation. Total time taken in this phase is 75 to 95%. Let us consider about the human cell. If we are considering about the human cell, the total time taken is 24 hours for the formation of the new cell. Then what time it will take in the interphase or intermitosis? The time taken here will be 23 hours. So you can understand the thing is 24 hours completes in interphase. Interface. Why? Because it is the preparatory phase. The whole preparation will take place in the interphase. And the actual division phase, that is the M phase, where actual division will taking place. As you are preparation for the NEET exam, the preparation will go on for a year. And now exam for a whole day or three and a half hours, it will take only that. So that's why the preparation is the major thing. So that's why it is taking so much high time. 75 to 95% of the generation time. Okay, so what is interphase? Intermitosis, time taking between the M phase and energy phase. It is total time taken 75 to 95% of the generation time. Now, this interface is further divided into G1 phase, means gap 1 phase, S phase, and G2 phase. Three stages will be under the interphase stage. Three stages will be here. G1 phase, yani means gap 1 phase, S phase, synthetic phase, G2 phase means gap 2 phase. So what will be the changes genetically, series of changes, or genetically series of changes taking place in gap 1, synthetic phase, and gap 2 phase? So let us discuss about these. Here, first one is gap 1 phase or G1 phase. Here, the amino acids, nucleotides, amino acids, nucleotides, ATPs, RNAs, and protein. Synthesis will take place in this phase. Why? Because here in synthetic phase, some constituents will be formed. What will be formed? We will discuss later. So the preparation of this phase will take in place in this phase. For this, the preparation will take in place. So requirement is amino acids, nucleotides, ATP, RNAs, and protein synthesis will take in place in this phase. This is the whole thing that will happen in the G1 phase. What protein? Means the raw material. Whole protein will synthesize here. That is histone protein synthesis. Other proteins also. But the raw material for that, the raw material that is required for this phase will be synthesized here. The preparatory phase for S phase is gap 1 phase where amino acids, nucleotides, ATP, RNAs, protein and synthesis taking place here. So what about the synthetic phase? The synthesis, histone synthesis will take in place. DNA duplication will take in place in this phase. Chromosome replication will take in place in this phase. Okay, and in case of animal cell, centriole replication will take in place. So you got in my point? Whole these synthesis will take in place in the S phase. The preparation for this will take in place in the gap 1 phase. So like this, the S phase is completed. Now the cell enters in the G2 phase. Which one? G2 phase. What will happen in the G2? Write it down here. Here the protein synthesis, RNA synthesis and macromolecules. So protein synthesis like tubulin proteins. Though that proteins is important in the spindle fiber formation. So protein synthesis, RNAs, those are required here for the protein synthesis, and macromolecules, cell organelles will be duplicate here. So whole thing will happen in these series of changes, that is gap 1, synthetic phase and G2 phase. Now the cell enters in the M phase. What will happen in the M phase? Why M phase is so important? Now M phase is actual divisional phase, and here the cell will be divided into two cells. This M phase is further divided into two stages, that is division in the nucleus and division in the cytoplasm. So what is the division in the nucleus? Means the nucleus will divide to form two nucleus, and again the cytoplasm will also divide. So the formation of the whole cell will take in place. Now two cells are ready. So write it down here. Write it down here. M phase. Already divided constituents or cell organelles will divide here. Divide. further divided into karyokinesis and cytokinesis. Karyokinesis means division in the nucleus, division in the cytoplasm, depending upon the cell. Here, whole cell is divided now. Now, what will happen when cell is ready? Cell is ready to divide again, then it will enter into G. When it is not ready, then it will enter into the G naught stage. It will rest, inactivation phase. And G naught phase is also termed as quiescent stage. What we call is G naught phase, quiescent stage. Quiescent stage. Here, division will not take in place, differentiation will take place. Cell matures and attain a particular shape and perform a different function. For example, nerve cell. It will never go in the G1 phase again. So that will happen like this. Now, the thing is, if we draw a diagram of the cell cycle, then how will it form for a human cell? See, so the revision will also take in place and the actual form will come like this for the human cell cycle. So I have told you the generation time is 24 hours. See here, this is the cell. Here, this is the cell. Here, this is M phase. This is G1. This is G1 S, G2 and M phase. Here, see what will happen in G1. RNA, protein, RNA, protein, nucleotides, amino acids, ATP. It will increase. Here, what will happen? Histone synthesis, chromosome duplication, DNA, all will synthesize here. And centriole. Now, the DNA content will be doubled in the synthetic phase. So, in case of haploid cell, DNA content previously it was 1C, it will be 2C. In case of diploid cell, it will be from 2C to 4C. But the chromosome number will remain the same. How? Like this, sister chromatid formation will take in place. Like this is chromatin fiber, and here sister chromatid formation will take place like this. This is sister chromatid. Write it down here. I am writing here chromatin. Okay, so the number, like in case of chromosome number is 1n, then it will be 1n. 1n. In case of diploid cell, chromosome number is 2n, then it will be 2n in the synthetic phase. S. This will happen. In case of G2, RNA, protein, and macromolecules. Synthesized here. Further, M phase here divided like prophase, metaphase, metaphase, anaphase, and telophase. This will happen in the cytokinesis. Sorry, karyokinesis. And again cytokinesis. The time interval, what will be decide like this? Here to here in interphase, that will take 23 hours. Here 1 hours only. Here it will be 10 hours. It will be 9 hours, 4 hours, and here 1 hour already written. So like this, total stages will completed. Example for 24 hours I have taken in culture form in case of human cell. So it is all about the cell cycle. I want to know from you the one question that is most important. What will happen in case of the autonomous organelles like and mitochondria and chloroplast? When they will divide, means the duplication here, here, or here. I have told you in macromolecules, but a autonomous means what we call mitochondria and chloroplast. The division in the chloroplast and mitochondria will take in place in the stage of interphase. You have to tell me in the comment box. Please comment me, and this is a very good question for you and for the AIMS level. From the best of my knowledge, I have told you everything. Thank you. Bye bye. We will meet you again in the next video with the detailed information about that, and the next video will be the what we called it, that the cell cycle completed, then it will be division. So division means mitosis. We will start in the next video. Till then, thank you. Bye bye.